Hello students, in this presentation we discuss about the mitochondria and chloroplast. The cytoplasm shows several membrane bound structure called cell organelles, which perform different functions and keep the cell in a dynamic state. Observations made with the help of electron microscope and other sophisticated techniques reveal the presence of plastids, mitochondria, endoplasm reticulum, ribosomes, Golgi complex, lysosomes, peroxisomes, glycosomes, etc. Now we will discuss about the mitochondria. As we have already know that what is the structure of the mitochondria, what are the parts are present in the mitochondria in your previous in inter level or in your high school level. Now we will discuss briefly about the mitochondria. These are the we can see in this diagram also. This is the structure of the mitochondria. In this then other one, these are the mitochondria, the rod shaped or spherical organelles which are concerned with the oxidation of 1 minute. These are the rod-shaped spherical organelles which are concerned with the oxidation of food materials. That process is called the respiration. They convert potential energy into kinetic energy and store it in the form of ATP. ATP means adenosine triphosphate. Adenosine triphosphate. Okay. They were first discovered by Kolliker. Altman discovered in 1894 described them as bioplast. And Benda in 1897 named them as mitochondria. Altman used it as the bioplast, but Benda named them as mitochondria. Okay. Mitochondria occurs in eukaryotic cells and their number varies from cell to cell depending on the functions occurring in cells. Usually they are found in a large number in actively dividing and growing cells. Okay. Then mitochondria measures about 7 micron in length. This is the micron. Mitochondria measures about 7 micron in length and 0.5 micron in width. Okay, in 7 micron in length and 0.5 micron in width. Okay, they possess double membrane envelope in which the unit membrane are separated by perimitochondrial space. So, this is the outer layer, outer membrane. This is one is the outer membrane. This one is the inner membrane. In between, perimitochondrial space is present in these two layers in between. So, the possess double membrane envelope in which the unit membrane, this is the unit membranes, are separated by a perimitochondrial space. This is the perimitochondrial space. We can see this in between the two layers place is called the perimitochondrial space. The outer membrane is smooth Outer membrane is smooth. This is the outer membrane. Smooth, but the inner membrane shows in invasionations. So these are the like structures. This is the inner membrane. You can see folding like structures. These all are the inner membrane structure. So the outer membrane is smooth, but the inner membrane shows invasionations called cristae. This part is called the cristae. This part is the cristae. You can see in this diagram. These are the cristae part in folding structures. Okay, the central space is filled with the fluid matrix. This is the central. This one is the fluid matrix. These are the fluid matrix. Okay, which consist of 70S ribosomes. Which consist of 70S ribosome. Which type of ribosomes is present? 70S type of. But in the prokaryotic cells, they have only 70S. But in eukaryotic cell, they have 80S and 70S ribosomes. But in the cytoplasm, 80S type of ribosomes present. But in the mitochondria matrix, they have 70S type of ribosomes. And circular DNA molecules and RNA. And which type of DNA? Circular DNA molecules. And which type of ribosomes present? 70S type ribosomes present. This is the 70S. Circular DNA molecules and RNA present. The matrix also comprises of respiratory enzymes in the matrix. They have respiratory enzymes that cause breakdown of food molecules by aerobic respiration. This process, it is called the respiration. Okay. The surface of cristae. Then we will go for this is the electron micrograph photograph mitochondria. This one is a three-dimensional layer. This is the matrix. This one is the outer layer. This one is the inner membrane. Inner member folds is called the cristae. Okay. In other diagram we can see these are the cristae. These are the cristae. In the surface of cristae is studded with tiny stalked particles known as oxysomes or F1 particles or F1 particles. These all are the F1 particles. Okay, in the diagram, this part is the enlarged in below diagram. This is the cristae. These all are the foldings are the cristae. In that cristae, some stalked particles are present, that is called the oxysomes or F1 particles. These particles are of the size of 8.5 nanometer in length and are arranged at distance of 10 nanometer from one another. In between these F1 particles, how much distance? 10 nanometer from one another. And each mitochondrion approximately has about 10 to the power of 4 to 10 to the power of 5 oxysomes. So, each mitochondria approximately has how many? 10 to the power of 4 to 10 to the power of 5 oxysomes are present. In Krebs cycle of respiration occurs in the matrix. The matrix, which processes occurs? Krebs cycle of respiration occurs in the matrix, and the electron transport take place. Which place? Take place on the cristae. Electron transport occurs where it is an cristae, but Krebs cycle of respiration occurs in the mitochondrial matrix. Okay, the oxysomes secrete several respiratory enzymes and release them into the matrix. The energy released during oxidation is stored in the mitochondrial matrix as ATP. Okay, hence mitochondria are termed as powerhouse of cells. Powerhouse of cells or cell furnaces or biofurnaces. Mitochondria possess their own genetic material and are self-duplicating. Hence, they are also called the semi-autonomous organelles. What? Semi-autonomous organelles. Why? Mitochondria possess their own genetic materials, so and are self-duplicating occurs. Hence, they are called the semi-autonomous organelles. Okay, this is the mitochondrial structure. These all are the mitochondrial structure. Then we will discuss about the plastids. So, in plastids, why are plants green? Plant cells have green chloroplasts. All of you know about this. Then, why are plants green? Means the plant cells have green chloroplasts. You can see in this diagram, they are in green in color. These are the internal structure of the chloroplast. These are the three-dimensional chloroplast structure. Then we will discuss in this. This is the color of light seen is the color not observed. So this is the light, observed light, transmitted light. This is one is the chloroplast, reflecting light. Okay, so chloroplast absorb light energy and converted it into chemical energy. The location and structure of chloroplast, where it is located. So in the this is the leaf structure. This one is the leaf cross section. This is the anatomical structure of the leaf. And this is the mesophyll cells. Okay, these are the three-dimensional chloroplast structure. Then we will discuss about the history of the plastids. First reported by Leeuwenhoek in 1698. Okay, but Schimper in 1883 named them as plastids. Named them as plastids. Okay, they are characteristic of plant cells and are generally absent in bacteria and fungi. Okay, they are arise from proplastids, which they arise from proplastids, but absent in the bacteria and fungi. They are characteristics of plant cells and are generally absent in bacteria and fungi. They arise from proplastids. Okay, the plastids are very variously pigmented and based on the pigmentation. Based on pigmentation, they are classified into three types, that is the leucoplastids, leucoplasts and chromoplast and chloroplasts, which they are plastids are variously pigmented and based on the pigmentation they are classified into three types: leucoplasts, chromoplast, chloroplast. Okay, then we will discuss about the each one. What is the amyloplast? That means also leucoplast. The colorless plastids are which are concerned with the storage of organic food material are called the leucoplasts. Okay, they are found in deeper tissues which are not exposed to sunlight. Okay, depending on the nature of food storage in them, they are identified as amyloplast, storing starch, aleuroplast, storing proteins, and elioplast, storing fats and oils. Okay, and depending on the nature of food stored in them, they are identified as amyloplasts. Hmm? And amyloplast, aleuroplast, elioplasts. In amyloplast, storing food is the starch. In the aleuroplasts, storing proteins, and elioplast, storing fats and oils. Okay. Then, uh, what about the chromoplast? Chromo means color. Plastids with orange, yellow, and red pigmentation are known as chromoplasts. Okay, they occur in the cells of petals, pericarp, leaves, seed coats, tubers, roots. They help in the pollination of flowers and dispersal of fruits and seeds. Chromoplast contains pigments called carotenoids, which are orange in red, that means carotenes, and yellow, that means xanthophylls in color. Roots of carrot have carotenes. Roots of carrot have carotenes. Fruits of tomato have lycopenes. All of you know about this. Okay. Red algae possess phycoerythrin, red pigment. That means red pigment. Blue-green algae contains phycocyanin. That means blue-green pigment. And brown algae show fucoxanthin. That is brown pigment. Okay. This is about the chromoplast. Then coming to the chloroplast. Chloro means green. They are green colored plastids. They are found in all green parts of plants like leaves, young branches, stems, sepals, unripe fruits, and expansionally in the roots of certain plants like Tinophyllum. They have chloroplasts. All autotrophic plants and chloroenchymatous cells possess chloroplast. They have a significant physiological role to play in cell metabolism, as they are chiefly concerned with the assimilation of food materials by photosynthesis process. Chloroplasts are spherical in shape, okay, or oval or discoid in shape. They measures about 5 to 10 micron in diameter. Okay, this is a three-dimensional structure of the. So, in plant cell about 20 to 40 chloroplasts are generally found. Each chloroplast is bounded by an envelope which is made up of two unit membranes that are separated by periplastidial space. We can see in this diagram also. This is the outer membrane. This one is the outer membrane. This is the inner membrane. Okay, this one is the outer membrane. This one is the inner membrane. In between two layers, separated by the periplastidial space of about 10 nanometer width. How much? 10 nanometer. Periplastidial space. You can see in this diagram also. This one is the inner membrane space. This one is the outer membrane. This one is the inner membrane. This is the three-dimensional structure. This one is the black and white diagram in your book also. This one is the color on two-dimensional. So the inner space of chloroplast is filled with colorless homogeneous matrix called stroma. This one is the stroma. These all are the stroma. This one is the stroma. Okay. So the inner space of chloroplast filled with a colorless homogeneous matrix called the stroma. This is the stroma, okay, which contains several photosynthetic enzymes, circular DNA. In this also they have circular DNA. In mitochondria also they have DNA, circular DNA, and 70s type of ribosomes present. Which type of ribosome? 70s. In mitochondria also they have 70s. And a large number of lipoprotein membrane extend from one end to the other and appear as a parallelly arranged lamella. You can see this is the a large number of lipoprotein membranes. This one is the lipoprotein membrane extend from one end to the another end to the other and appear as a parallel arranged. This is called the lamella. These are called the thylakoid also, or Fried membranes. Inside the thylakoids, a narrow lumen present, that is the empty space is present. In this we can see in between the two lamella they have lumen, empty space is present. At some place the thylakoids are arranged one above the other and appear as a stack of discoid membrane. We can see these are the at some places the thylakoids are arranged in one above the other and appear as a stack of discoid. We can see this one is the okay. So these are called the grana. We can see this one is the grana. In this also we can grana. Okay. In this diagram also. So chlorophyll is found in the thylakoid membranes. Thylakoid membranes are one quantosome, that is the 230 chlorophyll molecules and plus 40 to 50 carotenoids. Thylakoids function in the steps of photosynthesis that initially convert light energy to chemical energy. And stroma, photosynthetic reactions that convert carbon dioxide to sugar occur in the stroma. This is the stroma structure. These all are the stroma. And the grana, that is the singular, the granum. Stacks of thylakoids in a chloroplast. These are the grana structures. Okay. Then the chief pigment of chloroplast is chlorophyll. Other accessory pigments such as carotenoids are present along with chlorophyll. These pigments molecules are situated in the membrane of thylakoids. In the chloroplast, light reaction of photosynthesis occurs in the grana. So, in the grana, which reactions occur? These pigments molecules are situated in the membrane of thylakoid. In the chloroplast, light reactions of photosynthesis occur in the grana. In this, light reactions occurs, but in the dark reaction in the stroma. So, light reactions occurs in the granum, light reactions occurs in the stroma. Where it is? In the stroma, which reactions occurs? Light reaction. So, the photosynthesis, two stages are there. One is the light reaction and another one is the dark reaction. Light reactions occurs in the stroma and the dark reactions occurs in the granum. Okay. The presence of DNA in chloroplasts help in self-duplication in the mitochondria also. Hence, they are called semi-autonomous organelles. Okay. In view of this, some scientists described chloroplast is a cell within a cell or prokaryotic cell in a eukaryotic cell. Okay, so in the will be, this one is the not clear diagram, this is the A and B. Then carotenoids divided into carotenes and xanthophylls. Okay, then what is the function of the chlorophyll, chloroplast? And this is that they are the concerned with the manufacture of carbohydrates. All of you know about this, and sugars and starch through photosynthesis. Plastids change their color from one type to another as a result of growth changes taking place in a plant pots. In potato, when the tubers are exposed to air, leucoplast converted into chloroplast. In tomato and capsicum, the ovaries containing leucoplast which change into chloroplast after fertilization, and in ripe condition the chloroplasts are transformed into chromoplasts. So tomorrow we will discuss about the cell wall. Okay, thank you.